The overall goal of this salivary antibody bead-based multiplex immunoassay is to identify the etiological agents responsible for environmentally associated symptomatic and asymptomatic illnesses by measuring the IgG antibody responses to multiple pathogens simultaneously.
This method can help answer key questions in the field of exposure science. For example, which pathogens are responsible for causing illnesses in particular populations?
The main advantage of this technique is that it can be used to quickly and cost-effectively measure antibodies to multiple analytes at one time using very small sample volumes.
This method can provide insight to etiological agents and their sources of exposure, including foodborne, waterborne, and airborne pathways.
Visual demonstration of this procedure is critical because if you don't set it up properly, the results can be unreliable.
Begin by vortexing and sonicating the stock mixtures of beads coupled to the antigens of interest for 20 seconds at a time to generate homogeneous bead suspensions. Then dilute the coupled bead stocks to a final concentration of 100 beads per microliter of each unique bead set in PBS supplemented with 1% BSA. Next, set up at least 7 two-fold serial dilutions of anti-species IgG primary antibody in PBS plus BSA in a 96-well round bottom plate according to the manufacturer's recommendations. Pre-wet a separate 8-well column of a 96-well filter bottom plate for each antigen coupling confirmation test with 100 microliters of wash buffer, and remove the supernatant by vacuum.